All right. So a little bit of context of why do we think that machine learning and artificial intelligence can be useful for drug discovery. So I would say that the advantage or the main advantage of machine learning and artificial intelligence is that it's a very cost and time effective tool. It's experiments that you can do with your computer. Therefore, it's easy and quick to get results, and is much cheaper compared to actual wet lab experiments. Yeah, this means in drug discovery, where we have this complex pipeline that Jessica was describing so nicely yesterday, this means that you can fail faster by doing these machine learning approaches, and therefore hopefully save a lot of unnecessary expenditure. Another good thing about machine learning, especially for an initiative like ours, where we actually try to have many collaborations across the globe, is that we can do it remotely most of the time, which is quite convenient in this crazy world that we're living in now. And also an interesting thing, especially if you are like a data scientist, is that this field of machine learning is quite domain agnostic, which means that we can inherit progress from other fields like computer vision, image recognition, natural language processing, all of these things that receive heavy investment from large companies. Okay. Why do we believe that machine learning can be primarily applied to drug discovery, and especially to small molecule drug discovery in the context of infectious diseases in particular? Well, first of all, because we do have like all of the team here and the facilitators, we do have a track record in this field, and therefore it's easier for us to actually make connections between the data science field and the experimental results. That makes sense, I believe. Also, small molecules, as you know, are perhaps and will continue to be one of the best tools to tackle neglected and infectious diseases, because they are relatively cheap to produce and they are relatively easy to store. Yeah. There are, as you know, some inspiring initiatives in the field of antimicrobial drug discovery, for instance, medicine for malaria, drugs for neglected disease initiative, open source malaria, etc., etc. So we have good models of, let's say, initiatives that are trying to develop small molecules against infectious diseases, and we believe they are generating a lot of data, and we believe that machine learning can help there. Okay, so overall, the promise of machine learning applied to drug discovery. Jessica was mentioning yesterday how difficult it was with the traditional approach to actually get to an approved drug. And she was also explaining that the drug discovery pipeline starts with a collection of compounds from which you want to select one that is eventually good. The hope or the promise of machine learning is that we will be able to reduce the time to discover a drug, therefore decreasing costs, by increasing the number of molecules that we can consider computationally, right? So that's a bit what we are going to do, like during the sessions today, try to use these tools to actually screen, without doing any experiments, the compounds that look more promising, just by using the computer. So let's get into the topic, actually. So when it comes to machine learning, I couldn't come up with an easier way of actually defining the field broadly. Yeah, we have three families of algorithms, and I think that you need to remember this. This is perhaps the most important slide of the day. We have on one side what we're going to talk about today, which is supervised machine learning. By supervised machine learning, basically in this kind of family of algorithms, what we're trying to do is to classify or predict a certain label or activity. Yeah, you can see here in the plot, basically a machine learning algorithm would be this dashed line here that is trying to separate between, say, active and inactive compounds. Yeah, this is called supervised machine learning because you train those methods based on already known data so that you can identify this one. You supervise the method. Yeah. Yesterday, Jason very nicely explained another set of algorithms that are called, broadly speaking, unsupervised machine learning algorithms. And I think that the most popular type of method methods in this family are clustering methods, where you try to discover families or groups of entities, in this case compounds that look alike, and 2D projections, where you try to go from a very complex representation of, for instance, a compound to something that you can actually visualize. Similarity searches also fall within this category of unsupervised machine learning. Why do we call them unsupervised? Because you actually don't need information about the activity of those compounds to obtain clusters or projections, right? The only thing you need is the structure of the compounds. This is what we saw yesterday. There's a third family of methods, more complex, but also very interesting, which are called reinforcement learning methods. And in reinforcement learning methods, I think that the idea that you need to think about is like a robot. Yeah, so those are methods that that basically have an algorithm that interacts with an environment. And when it comes to drug discovery, I think that the most interesting application is generative models to invent new molecules. So why do we call this reinforcement learning? Well, because there's a method, an algorithm that generates a candidate, and there's an environment, for instance, a chemist or another algorithm that says whether this candidate is valid or not. If the candidate is valid, for instance, if the candidate that the network has generated looks interesting, then the network gets rewarded, and you start cycling and cycling and cycling. By doing this reinforcement cycling, you are able to generate hopefully compounds that look interesting overall. Yeah, this we will see on Friday just a little bit, because those are the most difficult algorithms to implement. Okay. Today is the turn of supervised methods, which I believe are the ones that you will end up using the most in your daily life. So how do we start, or how do we define a supervised method based on your experiments in the lab? Well, the easiest thing to do would be to think about active and inactive compounds. And how do we define active and inactive compounds based on experimental data? You know it better than I. We have, for instance, a 96-well plate. We have different compounds here. We have a dose-dependent experiment. If the bacteria, in this case, E. coli grows, then we have it purple. If it doesn't grow, we have it white. So if we overall see white, this means that the compound is active. If we see purple, it means that the compound is inactive, right? So by basically digesting this experiment and labeling these drugs, we can actually assign an activity value to each of the drugs. Yeah. So the next step would be to try and define a machine learning algorithm based on this data. So things start to get complicated now. We have structures on one side, and we have the activities on the other side. Yeah. So the first structure is active. The second is inactive. The third is active again. The fourth is inactive. Yeah. So the way we build almost always a machine learning algorithm that is able to map those structures to the activity value is by a two-step procedure. The first step, I call it featurization. Yeah. And in the featurization, we are basically doing what Jason was explaining yesterday a little bit, which is converting these chemical structures that you can draw as chemists into a numerical vector that the computer can understand. Okay? This procedure is the key, in my opinion, to successful machine learning. Once we have these numerical representations of the molecules, then it's just a matter of fitting a conventional algorithm, there's many of them, that are able to map these numbers to the outcome number. Yeah, so it's just, if you want, a correlation between the input numbers and the output numbers. Yeah. So this is where most of the machine learning research happens. But in my opinion, when it comes to drug discovery, here is where the most interesting part happens. Yeah. So I will say more time here than actually discussing the second part. Okay. All right. So why do we do all of this crazy algorithm? Well, because when you come with a new molecule for which you don't know the activity, the first thing you do is you featherize it using the same method that you used before. And because you have learned the connections between the input and the output, then you can make a prediction based on these features about this molecule. In this case, I predict that this molecule is going to be okay. So that's the whole concept of supervised machine learning, and it can get very, very complicated, but at the end of the day, we're always doing the same thing, okay? All right. So let's get started with featurization, which, as I said, in my opinion, is the most important and interesting step. So featurization basically means trying to identify in the molecule numerical features that are discriminative for the activity that we want to predict. So here I'm putting just a qualitative example that I came up with, but of course, as you will see, things get much more complicated. Let's say that we define, for instance, three features. One is the number of rings, in this case is 3. The other is the solubility, this is something that we can calculate. The third one is the number of fluors, in this case is 1. And the fourth number is the molecular weight. Yeah. And then we can do the following. We can plot these features, for instance, F1 versus F2, and see whether these features are able to discriminate between active and inactive compounds. Yeah, so I'm seeing here that F2 is kind of capable of doing so. I'm seeing here that F4 is kind of capable of doing so. And therefore, when I combine F2 and F4, I get a very good discriminative power. Yeah, so intuitively speaking, I have identified 2 features, F2 and F4, that if I use them smartly, we'll be able to tell apart active from inactive compounds. Yeah, so that is a successful featurization step. If you do featurization right, as I said before, the machine learning part is very easy. In this case, it's just drawing a line. Yeah. So, of course, life is not as easy. Usually, it's very difficult or even impossible to come up with an ideal set of features. So, what we do as modelers most of the time is actually doing like a gunshot approach. We define many features, and then we hope that some of them will be useful. And this is where the concept of chemical fingerprints that you are all familiar with come into play. So just to simplify a lot, what is a chemical fingerprint? A chemical fingerprint is a vectorial representation of a chemical structure, so a set of features that we can define by enumerating substructures or chemical patterns that we find in this molecule, right? And there's plenty, dozens or even maybe hundreds of types of fingerprints that you can find out there. One possibility would be to actually look at the structure and have a binary vector, with zeros and ones, and basically map every type of bond that I find in this structure into this vector. So, for instance, there's a position in the vector that is allocated for carbon and fluor. So if I find a carbon and fluor, which is the case, then I put a 1 here. Carbon and chloride, a 1 here. Hydroxyl here. Tertiary nitrogen here, etc., right? So I'm just looking at the molecule, finding all of the chemical features that I can find and map them in a vector. Yeah. Is this good? Well, it can be good. Maybe sometimes it's not enough, but for most of the purposes, most of the times it's good. This is also what we use, as you know, to do similarity searches between compounds using the Tanimoto similarity. Yeah. The good thing about this is that it's just a very simple vector, zeros and ones, and we know what each position corresponds to. Yeah. So just by looking at this vector, in principle, I would be able to kind of reconstruct the chemical structure, which is interesting. Yeah. Okay. So this is what I call classical fingerprints or descriptors. But of course, we are not in the big data age, right? So we know much more about compounds than just the chemical structure. Actually, if I go to the internet and I do a quick search, about that compound that I was showing before. I don't know if any of you noticed it, but this compound is called alophentrine. It belongs to a family of compounds called phenanthrines. It has been suggested to be an antimalarial compound. We even know the mechanism of action, blood-specific side. We even know that it has some off-targets in the host, and we know that those off-targets can cause, like for instance, cardiotoxicity. Yeah. So actually, to be honest, chemical structure is not the only thing about that we know about many of the compounds. Yeah. Gemma? Yes.
So my view was how are enantiomers and other isomers, same molecular formula but different spatial orientations, handled with fingerprinting, and how does the technique distinguish between two compounds in the same chemical groups?
Yeah. So enantiomers and stereochemistry in general, it's a difficult thing to capture with fingerprints. There are 3D fingerprints, which are a bit more difficult to explain, that are able to capture these sample bits. But in general terms, they are not successfully captured by many of these descriptors, the standard ones at least. Yeah. Good question.
Can you assign the features to the pharmacophore?
Yes. Yes. So pharmacophores, as you may know, are a kind of abstract representations of physical, chemical, and geometric features that you want for a particular activity. Yes, there are pharmacophore-based fingerprints as well. As I said, there's dozens of different types of fingerprints, and some of them are more pharmacophore-based, some of them are more topology-based, as the one that I was describing. Some of them are more 3D shape-based. Yeah.
You can also break them out. During categorization, how many features at least are considered enough?
So, good question. I think that at least I never use less than 500. Okay, that would be a good rule of thumb for most of the exercises I do. Never less than 500.
Does it identify the position of the bound between the molecule?
The position of the?
Bound.
Oh. The position of the molecular bond?
Yeah. Surprise, maybe.
Yeah.
Yes, I wanted to identify the position of the bound, knowing that when you have a finger sort of molecule, we have some bound which deny and some bound which one of the molecules. Taking account the chemical,
Yeah. The more complex and the more advanced the fingerprints, the more you can capture this kind of things, as in the case of stereochemistry, for instance. Yeah. Most of the conventional ones, they don't. And they are just simple enumerations of the structural features I will find, which might sound not enough, but it's surprisingly good in most of the scenarios. So I would recommend to go with the simple standard ones unless you have strong reasons not to do so. Yeah.
My next question maybe about the survey.
Yeah.
I was going to ask as well. So like for that, for instance, in this compound, you have 2 chlorines and 3 chlorines, and that's more than one. So does the fingerprint account for multiple, or is it just like the chlorine in the molecule?
Depends. There are fingerprints that are binary. In those cases, you have collisions. So it's the same having one than multiple, but there's many fingerprints that are count based, right? So if you find like 3 hydroxyls, you will have a 3. Yeah. All right. So let me continue. So I was saying we know much more about many molecules than just the chemical structure. And actually, at some point in my career, I wondered whether it would be possible to, by using machine learning, to actually capture all of this information into a format that is similar to what we have been using forever for describing small molecules, right? So the idea would be to actually go to the public domain, try to capture all of this information and convert it in a numerical format that is more or less similar or comparable to the conventional difference or descriptors. Yeah. And this is what I call the chemical checker. Is a resource that we published in Nature Biotech last year or 2 years ago. So to summarize the families or the types of fingerprints that we can find in general for small molecules, and then that we can use downstream for machine learning. We have, if we go to the classical type of fingerprints, we have 0D fingerprints. In this case, we call it them 0 dimensions because those are like properties like molecular weight, counts of nitrogens, things like that. Yeah, we have 1D fingerprints. Those would be the ones that I was just showing, where you just enumerate what you find in a structure. We have 2D fingerprints that actually capture a little bit better the shape of the compound on the 2D, like in the drawing. We have 3D fingerprints that are relevant to capture, for instance, stereochemistry issues. And we even have 4D fingerprints, which capture not only the shape of the compound, but also the, for instance, electrostatic characteristics of the different regions of this shape. Yeah. So this is what classically we have been using as inputs for machine learning. Of course, the more we go here in this direction, the more complex it is to calculate those fingerprints and the more elaborate those fingerprints are. Yeah. Whereas those are relatively easy to calculate, this can be quite complex because, as we know, we have multiple conformers. Calculating these electrostatic properties might be difficult, etc. Yeah. Now that we have data, a lot of data out there, we can actually come up with other types of fingerprints that I call data-driven. And basically, the idea of these fingerprints are actually to consider many compounds and not assume many things about them, just show them to a algorithm. And by just showing them to an algorithm, this algorithm hopefully is going to learn the relevant patterns in these compounds. Yeah, those are we call them autoencoder fingerprints. You will find them a lot in the literature, in the recent literature. Yeah, data-driven descriptors. Or we can even go taking the chemical checker approach that I was just describing. We can even go to the public domain, try to capture not only chemical information about the compound, but also the targets, biological processes, cell lines that might be affected, even clinical outcomes of these compounds, and try to convert them also in numerical format, as I was describing. Yeah. So we have all of this variety of fingerprints that we can use as input. The smarter we are, and the more we understand our problem, the better we can choose the best fingerprint that is going to yield the best machine learning model possible for our case. Yeah. Okay. So back to the chemical checker, just to simplify and just to give you a real snapshot of this database. This is how the chemical checker database looks like. Yeah, we have in each row here a molecule, and each molecule, because we are talking about fingerprints, is represented by a set of numbers. The classical solution, as we know, has been to represent just chemistry as a set of numbers. But thanks to the chemical checker approach, we can represent also the targets of the compounds, as we will see now, networks and biological processes, also cell-based assays, and also clinical outcomes of the compound. Yeah. So we go from having a relatively limited description of the compounds to having something that is very rich. And if it's very rich, then we have better chances of getting a good machine learning model out of it. Yeah. So just for you here, I think I'm showing about 100 compounds. I'm stratifying them in different therapeutic categories. And you can already see that there are some patterns that emerge, which means that these descriptors are likely informative of clinical outcomes, which is great. Yeah. Okay. So how do we build this huge database of abstract descriptors that are data-driven? Well, what we use is a type of database that we call it a graph database. And the graph databases are great because they are easy to explain. So, and to understand. In a graph database, we have a framework where we have nodes and edges. Each node here corresponds to a biological entity that I hope you can recognize. We have compounds or drugs, we have genes or proteins, we have diseases or phenotypes, we have cells, we have tissues, we have pathways where genes participate, etc. And then we have different type of edges connecting those entities. For instance, we know that compounds interact with proteins. We know that genes are associated with diseases. We know that compounds can treat diseases, but we also know that compounds can cause diseases. We know that genes are expressed in cells, etc., 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 right? So this is a great framework of biology. And all what we have to do now is to actually go to the public domain, screen a lot of databases and try to find evidence for these types of edges. Yeah. So this is what we do systematically, as you will see now in computational pipelines, so that we can populate this database like that. So how many edges? Each edge would be like an experimental point associated, for instance, a compound with a protein or a gene with a disease. How many edges do we have today? Today, in a database like that, we have about 110 million edges. So there's a lot of information that we have been gathering over the years. Here I'm just showing, I think it's 50,000. Yes, it's a very, very small extract of this database, just for credibility, let's say. Okay, so bottom line would be that thanks to this database, we have an integrated view of a lot of the pharmacological and biomedical evidence that we have been gathering in the public domain over the years. This is great because we can work not only on compounds, but we can also focus on proteins. So now I'm gonna detour a little bit, but I hope it's gonna make sense. Basically the same thing that we've been doing for compounds, trying to describe compounds in a numerical format, we can do it also with proteins. And so with proteins, we can take different types of descriptors or approaches to describe them. Some of them are sequence-based, basically the same idea. We just look at the sequence of the protein and we convert it into a numerical vector somehow. We can look at the sequence, let's say from an alignment point of view, but we can also look at the sequence as if it was like a sequence of words, like a sentence, and leverage natural language processing methods for actually understanding what's going on in that sequence. That's a very interesting family of algorithms. At the end of the day, the idea is the same. We look at the sequence, we end up with a set of numbers that describe the protein. Yeah, we can not look at the sequence, but also actually look at the structure. And in this case, we can come up with geometrical features or even with images from the structure that we can convert into numerical vectors. Yeah. So the same thing, exactly the same thing that we were doing with compounds, we can do with proteins, which is great because as you can see, we are converging into a common mathematical framework, which is just representing biological and pharmacological entities as sets of numbers. Yeah. We can even go more crazy than that and actually look not only at the sequence of the proteins, but as I was doing with the compounds, look at all the rest of the things that we know about these proteins, for instance, their participation or importance in cell lines, their relevance for disease, their participation in pathways, biological processes, etc., or their patterns of interaction with compounds. All of these we can capture, again, as sets of numbers. Yeah. So the idea is that whatever type of information we want to capture, we can convert it into numbers, and then those numbers can be used prospectively and as predictive features for machine learning. So all of this sounds very abstract, but it's actually the foundation of one of the most important, actually the breakthrough of last year, the medical breakthrough of last year, which I'm sure you are familiar with it, is called AlphaFold. History of AlphaFold is basically it's an algorithm developed by DeepMind, which is a company that is not necessarily expert in biomedicine, but they are experts in machine learning. Actually, one of the first breakthroughs of DeepMind was this AlphaGo algorithm. You might have seen the documentary on Netflix. It's just a neural network that was able to beat the best player in history in this quite difficult game called Go. So these people have applied their methods to protein sequences, basically, just by using these embedding techniques that I was, these descriptors that I was describing, so that now today, thanks to this AlphaFold method, just from an amino acid sequence, we are able to predict with striking accuracy the fold of the yeah. So actually, the same day AlphaFold was released, I was so curious about it, and just by using this laptop computer, I was able to resolve the structure of the target that I was interested in at the time. Okay, so just 1 hour of my time, I was able to resolve, hopefully with good accuracy, the structure of a target for which we didn't have extracted before. People at AlphaFold have gone like the extra mile of actually doing all of these pre-calculations over the last year for 200 million proteins. Yeah, so now today there's a database of pre-calculated and AlphaFold structures for essentially any protein sequence that you can think of. Yeah. So machine learning here in this case, in the domain of folding, has been instrumental in actually changing the field completely of structural biology. And the fundamental principle that goes behind is this descriptors principles that I was describing. All right, so now back to compounds, which is what we care about. So if we think about this huge database that we are collecting, basically the node that is most interesting to us are the compounds. Yeah, so as you know, there's a lot of data about compounds, a lot of data out there about compounds. So actually to populate the edges around this node, we have to do a lot of computing pipelines, yes, of bioinformatics pipelines, so that we can gather information from many different public databases. Yesterday, Jason was introducing Campbell, so Campbell is just a small thing here, so you can imagine how complicated things can get. Yeah? But long story short, at the end of the day, we can come up with a database of signatures or descriptors, as I was saying before, that is able to capture the chemical properties of the compounds, the targets that we know of these compounds, the biological processes and interactons and pathways that might be affected by those compounds, cell line effects in cell lines, and also clinical outcomes. And within these 5 levels, like broad 5 levels of increasing complexity, if you want, we have different sublevels. For instance, when it comes to chemistry, we might want to capture the 2D structure, but also the 3D structure or specific chemical properties. When it comes to targets, we might want to capture the intended mechanism of action, but also metabolic genes, for instance, or even off targets of the compounds. When it comes to networks, we might want to capture signaling pathways, but also like biological processes or localization in the cell. When it comes to cell, there's all sorts of types of data that we can collect today with phenotypic screening assays. For instance, we might want to capture changes in the transcriptomics of the compounds, or growth inhibition profiles across broad collections of cell lines, or even changes in morphology. When it comes to the clinic, we capture therapeutic areas or indications, so things that are good, but also side effects and toxicology. Okay. So by doing this systematically twice a year, it's a pipeline that runs over like 3 weeks or so. We're able to collect a lot of data. Today, I think we have, this is outdated, we have roughly 1 million compounds in the chemical checker data. So it's quite big. It is by far the largest collection of pre-calculated descriptors of small molecules that is data-driven, because we collect data from other sources. You can find it here, chemicalchecker.org or bioactivitysignatures.org. And of course, since we published this in 2020, we've been applying the tool in all sorts of methods and applications. This would be one example, and I think that one that we are particularly proud of is this Dream Challenge, where it was an open contest to predict kinase activity profiles of small molecules. And just by using the chemical checker and very, very simple machine learning algorithms, we actually were winning in this challenge. All right, so let's go back to concepts that we saw yesterday. You might remember these 2D projections, UMAP projections. So one of the nice things we can do with the chemical checker is that we can think of projections of different types. For instance, here, I'm showing projections that capture this corner here, which is the transcriptional response of molecules. Basically, what it means is that if two molecules are nearby in this projection, it means that when cell lines are exposed to those molecules, they upregulate and downregulate similar types of genes. Okay, similar genes, similar sets of genes. Cell sensitivity profiles, it means that if we assay these molecules against a collection of cells, hundreds of cells, for instance, cancer cell lines, they will tend to kill the same or inhibit the growth at least of the same types of cells. Yeah. Chemical structure, we saw it yesterday. So those maps capture whether molecules are similar or not chemically. So compounds that are nearby means that they are chemically similar. If they are far apart, it means that they are chemically dissimilar. And by having these three maps, we can do all sorts of interesting exercises. For instance, here, I'm looking for compounds that seem to have similar phenotypic effects in cells because they upregulate and downregulate the same sets of genes. They seem to kill the same types of cells. However, when we look at the chemical structure, they are completely different. Yeah, so that's quite interesting if you think about it from a drug discovery perspective, because this gives us a chance to identify compounds based on the phenotype, not based on just chemical similarity. So those compounds are, by definition, perhaps more novel. Actually, we identified a new molecule within these clusters for which we didn't have an annotation. And indeed, when we tested it, we realized that this molecule had the same mechanism of action as the rest of compounds, which was interacting in some capacity with DNA. Okay. We can do exactly the same thing, but the other way around. We can take two famous families of antimicrobials. You can see them here in orange and purple. We can plot them in the chemical map first. And indeed, all cephalosporins are nearby. All sulfonamides are nearby. They indeed have the same mechanism of action when we look at them in the map that corresponds to mechanism of action of the chemical checker. But when we look at the transcriptional profiles, that is the effect on human cell lines, transcriptional effect on human cell lines, we see a lot of sparsity, which might indicate that these compounds, even if they have very similar structures, even if they interact with the same targets in the microbe, they might have very different side effect profiles in the host. Okay. So those are the kind of exercise that we can do by using these data-driven descriptors. We can even do more interesting things. For instance, we can do an exercise where we are trying to identify mimetics of biological drugs. As you know, biological drugs are a very interesting family of drugs, but they have a lot of problems, especially in pharmacokinetics, right? And they are also expensive to manufacture. So, of course, one interesting thing to do from a drug discovery perspective would be to identify small molecules that might act similarly to a successful biological drug. Yeah. So in this case, we took an IL-2 receptor inhibitor antibody called the taclizumab, and we looked at the transcriptional profile of the taclizumab in cells. So we had an assay where we could collect what were the genes that were upregulated and downregulated when they were exposed to this antibody drug called the taclizumab. And then we can ask the following question. In my database, in my chemical checker database, can I find compounds that have a very similar transcriptional response to the taclizumab? Why do I want to do that? Because if they have similar transcriptional response, it is possible that they might act with a similar or related mechanism of action, right? Because they have the same phenotypic effect. So in this case, in this virtual screening case, we are identifying this would be a hit because it's similar to the glisomab. You see blue here and blue here, red here and red here, and this would not be a hit because it doesn't look like the glisomab. Yeah. So we did that for, at the time we had 10,000 compounds, and we came up with 19 hits computationally, which we purchased or synthesized and tested. And out of the 19, 14 worked to some degree. And by work, I mean they elicited a similar effect than the glisomab in the characteristic assay of the glisomab, which is IL-2 IL-2 dependent proliferation. As you can see here, for 4 of the selected compounds, there is a dose-dependent response that's comparable at least to the one of the glisomab. So that was a great success. Actually, when we had the closer inspection into this data, we realized that not all of the compounds seemed to act through the same mechanism of action, which kind of indicated that some of the compounds were not inhibiting the IL-2 receptor per se, as it is the case of the glisomab, but they were inhibiting kinases downstream the pathway. Yeah, so that was quite an interesting virtual screening exercise because we were discovering new families of compounds that could be interesting as mimetics of this biological drug. Okay, so I need to speed up now, okay, because I'm aware of time. So there's a huge problem about this approach, which is, as we talked about this yesterday, the chemical space is huge. It's actually unattainable, right? So, and when we think about available data, we don't have data for the vast majority of the compounds. Yeah, so actually, when we think about the chemical checker, the molecules that we have are not the full chemical space, but only a privileged region of it, the one for which we have data, which is extremely problematic for someone who tries to do modeling, because we encounter a problem which is very similar to the problem that the Romans had, and when they were trying to navigate the world, they had a very good or quite good view or notion of how the Mare Nostrum or the Mediterranean Sea looked like. Why? Because they had sampled it a lot. They had a lot of information. Sorry. However, when it comes to more interesting parts of the world, like where we are, they really had no idea. Yeah. So, and those are the interesting parts of the world because those are the new molecules, the molecules that we're inventing for which we don't have data, right? So what can we do about it? How can we get actually like information or some kind of inference for the molecules that for which we actually don't have any experimental data? Yeah. So actually a more honest view about the chemical checker is something like that. We always have chemical information, but when it comes, for instance, to collecting data about, let's say, the clinics, most of the times we know nothing about it. Yeah. So the second task we had when we were building the chemical checker was trying to fill in the resource, at least with predictions or some inferences where they were missing. Yeah, and as you can imagine, this was a huge computational task, huge challenge, because it's a lot of inferences that we have to do. And what we chose for doing so was a kind of algorithms called Chinese neural networks. Don't need to worry about it. Those are very interesting networks because what they do is they try to learn patterns of similarities between compounds. And the notion, intuitively speaking at least, would be that you learn how to compare molecules for which you miss data based on the data that you have. For instance, if I want to compare the side effects of compounds, but I don't know their side effects, basically I look at other spaces like gene expression. Perhaps I have data here, perhaps I have some binding data, and for sure I have chemical data. And based on the data that I can collect somewhere else, I try to make an inference here. Yeah. Long story short, building this network was a huge computational task. We used a supercomputer, which in my opinion is the most beautiful we have in the world. This one is in Barcelona. What's beautiful about this is that it's based inside the church, as you can see. So it's actually quite nice. And we were doing computations on exactly on those computers here. Yeah, at the end of the day, I think it was over a week of calculations. We came up with a network that basically allows us to do the following, Siamese neural network that allows us to do the following, even a compound and partial data about it, we are able to complete the missing data. Yeah. Of course, we are aware that this will not be perfect because it's a lot of predictions we are doing, but still we are able to somehow evaluate where do we have confidence about predictions and not. So for instance, when it comes to binding data, we were quite good at predicting it. When it comes to, for instance, transcriptional data, we're not as good at predicting it. But we still have some predictive capacity in all types of data. Actually, when we look at it in more detail, we were actually even able to identify regions of the chemical space for which we had more confidence. That would be the mare nostrum in blue, and regions of the chemical space for which we were less confident. That would be the outer part of the world, the regions for which we have less data. So yeah, so that's quite a nice framework because we can have, in principle, all of this information in principle for any molecule we can think of. So the question is, does it work? Well, I think it does in many scenarios. So for instance, here you can see again these 2D plots that Jason was so nicely presenting. In this case, I'm projecting molecules based on all of the information that I have been predicting. Yeah, so it's not only chemical similarity, but it's actually biological similarity. Yeah. And in the first row, I present compounds. Two compounds are similar if they are nearby, if they have similar chemical checker profiles. And as you can see here, I'm stratifying or highlighting compounds in different therapeutic categories. And you can see quite nicely and quite strikingly, I would say, that depending on the disease area, you find compounds in different regions of the chemical checker space, which is great if you think about it, because when you come with a new compound, and if the new compound falls here, you have good reasons to think that this compound will be useful in psychiatry, for instance. Yeah. Falls here, you have good reasons to think that it might be a good anti-infective compound, etc. This applies not only to drug molecules, but also to metabolites of different species, to metabolites inside our bodies, in different fluids, or even to food ingredients. Yeah, so these descriptors are hopefully very informative about the eventual activity of the molecules, and therefore are very good inputs, we believe, for machine learning tasks. All right, so I have exactly 5 minutes. I don't know, I can either stop here and we find another place to continue. I have like 5 or 6 slides left. What do you think? Maybe it's going to be 10 minutes altogether. What do you think? Should I continue? Do we all agree? Yeah? Yeah? Okay. All right. So we've done we've gone through the most difficult part already. Now it's just downhill, okay? Let's go back to our favorite scheme. We've covered, especially with the chemical checker, how to theaterize molecules. Now it's time to see how do we go from this abstract numerical representation that nobody understands of chemical compounds into an actual prediction of activity. Yeah, and for this we need to do some machine learning, some supervised machine learning that allows us to map from numbers to outcomes. Yeah. All right. So inside supervised machine learning, we have 2 different types of tasks. Second most important slide for today. We have classification tasks and regression tasks. Okay? Classification tasks, roughly speaking, active, inactive. Yeah? Green, active, process, inactive. We want to classify with an algorithm. This is the algorithm. We want to discriminate between active and inactive. Some people are not satisfied with that, and they would actually want to predict the actual, let's say, IC50 value. Yeah. In this case, you have to go for a regression, where you have experimental data, have predictions, and you want, of course, you want to make predictions so that they fit experimental data. In general terms, That's easy, that's more difficult. Yeah, so we will start, if we all agree, with the classification tasks, because they are usually enough most of the times. Even if nobody wants to put a cutoff, they are usually enough. Okay, so let's start with classification tasks today. And let's focus on the kind of challenges that we have when we are trying to do a classification. Because as I said, life is not as easy as it looks in those diagrams. We have 4 types of challenges, in my opinion, that I want to highlight, based on also in discussions from yesterday. First challenge, overfitting. Yeah. As I said, life is not easy. Sometimes it's not so easy to discriminate between actives and inactives. And because those machine learning methods are so powerful and those feature vectors, so those descriptors are so large, you can easily overfit. Yeah. Which means you draw a model that is too complex for your problem. Yeah. This is not good because when you come with new data, the model is not going to work so well. Okay. So this is one of the things that we need to control, and Jem is going to show afterwards how to do it. The second problem, we were calling it bias yesterday, but it's, if we think about classification, is class imbalance. Usually, typically speaking, experimentally, many times we have many inactives and just a few actives. Okay, so this might cause the model to become lazy and just call everything inactive, and it's going to be fine most of the time. Yeah, so this is not something you want to do. You have to avoid or correct for, we call it class imbalance. And you want to actually give more importance to the actives so that the model doesn't become lazy. Yeah. Third, very important concept about classification is the confidence or the applicability domain of the model. We were talking about it a little bit yesterday with Jason. Here in green, we have the known active in the training set. Here in the crosses, we have the inactives, and we've came up with a good model, simple enough model that is able to discriminate between the two. Now, when I come with new data in purple, I have 3 different scenarios at least. One where the prediction is very easy. This one, I'm very confident about it. It's going to be active. One where it's in the decision boundary, not so clear for me. And one where it's outside the applicability domain. I really don't know what happens here because I don't have training samples around it. Yeah. So knowing where I am, it's very important to actually assigning confidence to my predictions. Okay. Those are high confidence predictions. Those are so-so confidence predictions, and those are low confidence predictions. Yeah. Very important to quantify this. Finally, most difficult thing you can think of in machine learning is interpretability. Everything is super abstract. Everything is super complex. How do I map back to the structure? How do I know what features of the molecule are actually important? So I can talk to a chemist so that they can optimize their molecules to increase the activity. This is the most difficult thing to do. As modelers, we will not have time to actually do it in this course, but just remember this this area of machine learning called interpretability or transparency or explainability. It's an active area of research, and it's likely that you will find a lot of machine learning papers in drug discovery that are actually focusing on this, on mapping the important parts of the structure that confer the predictive power. Yeah. Okay. Last thing we need to do for machine learning, very important, is validating the models. And the way we typically validate the models, this is something that Jim is going to focus on, is by doing train test splits. Yeah. The idea is that we train the model only on a subset of the data, and then we evaluate it on the other subset of the data. Yeah. So we train on 80% of the data, for instance, we evaluate the model on 20% of the data, so that we have an idea of the predictive power of our model. If you have enough data, actually you want to do 3 splits, as we will see. Train, validation, and test. Why do we want to do that? Because we can use the train and validation to fine tune our model, so it works very well. And then we finally test with the yellow one, which is the test. Okay, we'll see this later on today, how to do that. It's not that difficult. Yeah. But this is very important. If you read the paper and you don't find, we call it cross-validation data, cross-validation results, don't trust that paper. It's very important that we assess the performance of the model by doing this kind of procedures. Yeah. All right. So at the end of the day, there's a lot of things we need to do as modelers. Thankfully, there's automated pipelines to do that. We will see a little bit how to do this easily. But at Ercilia, we've been developing some tools together with Jason to make this procedure very automatic. Yeah. And it works pretty well, I have to say. So here, I'm just showing a benchmark across different tasks. Some tasks are more biophysical, like interaction between different targets, or to base, which is a protein, or even to the HIV virus. And some tasks are more physiological, like clinical toxicity, side effects, etc. And as you can see here in blue, it's a method based on the chemical checker signatures that I was describing. Most of the times, if not always, the chemical checker gives us better predictive performance. Yeah, it's able to use the information that is available out there to actually have better starting points for the models. And this is especially true for the physiological tasks, the tasks that are more difficult or more advanced in the clinic, in the drug discovery pipeline. Let's focus very, very briefly on one of these tasks, the TOX21 in this case. This benchmark contains 12 toxicity pathways that are very important to monitor during the drug discovery process. And as you can see here in blue, there is the performance in the cross-validation of the chemical checker-based models. And here is the performance of the classical models based on just chemical structures. And as you can see, almost always, we have better results achieved by the chemical checker signatures, which is very promising. Not only this, but if we look at this small inset here, as we remove more and more data, so we simulate that we have less and less data, which is a topic that people are very worried about. What happens when we have just a few data points? The performance drops dramatically if we only use chemical information, but it kind of halts if we use these richer descriptors like the chemical checker descriptors. Yeah. So those results are, I believe, very promising in terms of modeling. This is why we have applied approaches like this one, as Jason was describing in HGD, to actually mimic or mirror the virtual screening cascade that they have in place with the experimental screening cascade that they have in place with the virtual screening cascade based on machine learning models that is able to actually predict the fate of the compounds across the cascade. And as Jason described nicely, we have whole cell assays for different pathogens. We also have toxicity assays, even some ADMET assays, and even some enzymology. Okay, by using the kind of supervised machine learning tools that I've just described. Yesterday, some people asked about transfer learning. Here I'm just showing a reference that you can check of a very good paper implementing transfer learning. The idea is, again, to use this kind of descriptors to actually learn from cell line assays and then fine-tune those models with clinical samples. Yeah, so we go from a large data set to a smaller data set that is very predictive. The pre-training with the large data set is essential to actually have good predictive power downstream. And this is a good article showing this in the realm of cancer. Yeah. Okay, so all of this, I hope it's useful, but it might be scary. So what we do at Ercilia is trying to bundle all of this procedure of like training the model, testing, tuning, validating, etc. In this outer circle, which we call deployment, so that actually, if you want to use the models, you don't have to do much. Everything we do it ourselves, and you can just use the models very simply with a few lines of code. I believe that, I'm going to skip this slide, that this can be interesting because it has the potential of bringing state-of-the-art machine learning research actually to the experimental lab. For instance, there's this Cell paper, 2020, presenting a deep learning approach for antimicrobial drug discovery, so broad spectrum antimicrobial activity. Very nice paper, very complex code. We bundle it within the Ercilia machinery so that researchers have an interface that looks like this. They can input the drug and they will get a prediction. Yeah, so something that was very scary becomes very easy, hopefully all of a sudden. Sometimes we don't have a machine learning model that the researchers have developed. So what we do is some training in-house, as I have described. For instance, we found this paper, 2018 in Science, large-scale screening campaign for antimalarial activity. We can collect that data from the supplementary materials, build the machine learning model, and deploy it in Ercilia so that a researcher can just come, input their molecule, and get a prediction, right? That reflects experimental screening assays that are underlying the model. Of course, what makes our life more fun is actually to collaborate with people. So what we really like is to actually engage with researchers that have data, experimental data, and build models for them so that we can come up with tools that are customized to their research. For instance, this would be an example in Cameroon. They have natural product activities. We can build models with that so that when they come with a new natural product, they can have some sort of prediction. Yeah, this is what we do as part of our mission. You can check our website, plenty of projects there, as you will see. And perhaps the most important or interesting one is this one, which is the Ercilia Model Hub, where we are gathering all of these models that Gemma is going to explain later on. So this is the website of Ercilia, and this is how the Model Hub looks like. Plenty of models. Our plan is to have 500 by the end of the year or so. At the moment, we have about 7. Yeah, but we are up to speed, so we believe we'll reach this objective by the end of the year. I'm done. Sorry, I was a bit delayed. So take-home messages from this talk. I think it's 6. Supervised machine learning is what you want to do for activity prediction. Supervised machine learning. Describing molecules correctly is the most important step. It's very important if you want to build good predictors downstream. So really think about your chemical space and how to better describe it. We present a way that we believe works in many ways, in many cases, which is the chemical checker, which are these kind of data-driven descriptors. I have explained some of the challenges of machine learning, supervised machine learning. The most important one, I believe, is overfitting. We have this tool called Ercilia Model Hub that we will see in the afternoon, which contains some pre-trained models already for you to use in predictive mode. And yeah, we'll see in the hands-on sessions how we actually use these models and make some interesting predictions for you for some of the compounds. Okay.